Hello friends, today we are going to discuss about phytohormone, which is an important topic of plant physiology. In this video, we will mainly focus on the auxin plant hormone. So first of all, let us see what is plant growth. It is irreversible change in size. That means the change in size is permanent or it cannot be reversed. And this change in size may be increase in length or volume. And this growth is considered as increase in dry weight. Wet weight is not considered because it keeps on changing. So how plant growth is controlled? These are controlled or regulated by phytohormones, which are also known as plant hormones or plant growth regulator. Let us see its definition. These plant hormones are naturally produced organic substances and these are produced in higher plants. These control the growth or physiological functions in minute amount. That means in order to control the growth or physiological functions, small amount of phytohormones are required. Now let us see the features of phytohormones. These are small organic compounds. These are synthesized by specific plant cells or tissues. These are active in low concentration. It means only small amount of phytohormones are required in order to control the plant growth. Promote or inhibit growth or developmental responses. Site of function is remote from site of production. That means the site where it shows the function is separate from the site where it is produced. Now the types of plant hormones. We will discuss about 5 major types of plant hormones. Auxins, gibberellins, cytokinins, abscisic acid and ethylene. Let us discuss them one by one. Auxin. Name is given from the Greek word auxin, that means to increase. It is the first plant hormone that was discovered. It was discovered in the last quarter of 19th century and it was successfully isolated by Fritz Wendt from the tip of Avena sativa coleoptiles, which is oats. Now let us discuss some of the experiments that were conducted which led to the discovery of auxin hormone. First of all, in 1880, Darwin did his experiment. He took the coleoptile of grass and exposed it with unilateral light. What he saw that these coleoptile bent towards the light. Later on, he covered the tip of the coleoptile with the tin foil and then exposed to the unilateral light. At this time, no curvature or bending was seen. So what it was concluded that tip of the coleoptile senses light and also that certain stimulus is transmitted from the tip of the coleoptile to the lower region, which causes the bending of the coleoptile in the direction of the light. But when the tip is covered with tin foil, light is not sensed. In 1910, Auxin and Jensen cut the tip of the coleoptile and then replaced it, keeping a gelatin plate, and was exposed with the unilateral light. Again, the bending of the coleoptile was seen. So what he concluded on the basis of which that the stimulus that Darwin has mentioned is actually material substance which diffuses through the gelatin from the tip of the coleoptile to the lower part. In 1919, Pal, he cut the tip of the coleoptile and placed it asymmetrically. Then it was kept in the dark, but still the coleoptile shows the curvature or a bend. So what it was concluded that these material substances that were produced from the tip, when these diffuses to the lower part, then it causes the growth of the lower part. Here This is placed asymmetrically, so these diffuses to the lower part or transmitted to the lower part only in this region. As a result of which this region grows more compared to this region. So there is asymmetry in the growth of these two regions. As a result of which this region grows more compared to this, and hence it bends or it shows the curvature. And this also explains the bending of the coleoptile in the direction of the light when it is exposed to unilateral light. Because in the presence of the unilateral light, there is more concentration of auxin in the shaded region compared to the illuminated region, and hence more growth or elongation of this region compared to this region, as a result of which there is asymmetric growth and coleoptile bend towards the direction of light, or the curvature is seen. But when light is given to the coleoptile equally in all direction, then there is no asymmetric growth, but the growth is symmetric, and hence in that case there is no bending of coleoptile or curvature is not seen. Later on in 1928, Wendt cut the tip of the coleoptile and placed them on the gelatin or agar block. Then these agar block were placed on this coleoptile asymmetrically, and then these were placed in dark. It was seen that this shows the curvature. That means the material substance which causes the growth of the lower part has diffused from the tip of the coleoptile to the agar block, and when it was placed, then it diffuses from agar block to the lower region. Since it is placed asymmetrically, this region receives this substance, but this region doesn't receive the substance. Hence, this region shows more growth compared to this, and this results in the bending or the curvature in the coleoptile. And this led to the discovery of Auxin, and on the basis of this experiment, Wendt was able to isolate Auxin from the Avena sativa coleoptile, that is the oats coleoptile. Some of the naturally occurring auxins are indole 3-acetic acid IAA. This is its chemical structure. Indole 3-butyric acid, that is IBA. This is its chemical structure. Phenyl acetic acid is also naturally occurring auxins. There are more than 200 auxin analogues also present, and these have the common feature, that is these have the unsaturated planar ring structure. This is the planar ring structure and a carboxyl side chain. This is carboxyl side chain. Now let us see some of the physiological effects of auxins. First is cell elongation. It stimulates elongation or expansion of cells in shoot. Cell division. It stimulates cell division in cambium and also in tissue culture with cytokinins. That is, in the tissue culture, in addition to cytokinins, when auxin is also added, then it results in the cell division. Cell differentiation. It stimulates cell differentiation of phloem and xylem. Root initiation. It stimulates root initiation on stem cuttings and also lateral root in tissue culture. Apical dominance. When there is apical bud present, it inhibits the growth of the lateral bud. This is known as apical dominance. When the apical bud is removed, then lateral buds grow. Flowering. It promotes flowering in bromeliads and femaleness in dioecious flowers. Next is callus formation. In many tissue culture, by the addition of auxin. So in many tissue culture, when auxin is added, it results in the formation of callus. What is callus? It is undifferentiated mass of cell. When auxin is added in the tissue culture, then it results in the division of the cell, and hence undifferentiated mass of cell is formed, which is known as callus formation. tropic response, that is bending to gravity known as geotropism and bending to light known as phototropism. This phototropism we have already seen in the experiments that were conducted in the discovery of auxin. Parthenocarpy. Auxin induce parthenocarpic fruits. What are parthenocarpic fruits? Fruits that are produced without fertilization of ovule. It occurs naturally but can be induced artificially. So this is all for today's video. In the next video, we will discuss about biosynthesis of auxin. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.